Mike. Welcome to the August Evening Skidaway program. Two quick announcements before I introduce our speaker. First off, if you are not on our mailing list, our summer newsletter, there's some copies here, there's some copies back on the table, and there's some copies in the lobby. And as always, in the newsletter, if you're not a member of our foundation, is a remittance envelope, and we always appreciate your generosity. Secondly, we are going to have a bonus Evening at Skidaway program 2 weeks from tonight. We were approached by the folks at Marine Extension long after we put out the annual schedule, if we could squeeze another program in. It's being presented by a woman by the name of Barbara Mann, who is a Athens-based metal artist, and she combines her art with marine science. Not exactly sure how that's going to work. I'm telling you, that's what they're telling me. And she's going to have a display at the aquarium. And she's going to come and talk about her work. And again, if you're on our email list, you should have received an email this afternoon giving you all the details. But the 22nd, 2 weeks from tonight, 6:30. Everything else is the same, but it's not on our regular schedule because this was a late add. I'll call it a bonus. Our speaker tonight, Dr. Natalie Cohen. Dr. Cohen received her bachelor's degree in biology from Penn State and then a PhD in marine sciences from the University of North Carolina. She joined us in 2020 following a postdoc fellowship at Woods Hole Oceanographic Institute. Her area of concentration is basically how the tiny little things react to changes in their environment on small scale, like molecular scale, up to ecological scales. And on that note, I will turn it over to Dr. Cohen.
Thank you so much, Mike, and thank you everyone for joining us tonight. I am Natalie. I am an assistant professor here. I've been here for about 2.5 years now. Tonight, I'm going to talk about harmful algal blooms, what they are, and their relevance in coastal Georgia. So here you're looking at our Skidaway River Estuary. This is a drone image taken by our director, Clark Alexander. And you can see our institute, Skidaway Institute, as well as Marine Extension in this photo. A little bit more about me. Mike just did a fantastic introduction. I started my academic journey a while ago. So I was an undergraduate student at Penn State, and I majored in biology. And there I was exposed to the world of oceanography. I had fantastic professors that really excited us about this newer field of science. I then continued on and obtained my PhD from the University of North Carolina at Chapel Hill in marine sciences. I was able to go on a lot of fieldwork expeditions, and these are some images of me in the field. I learned a ton about surviving on boats and conducting research. I also did quite a lot of work in the lab, and we do a lot of computer science work, which I'm not going to talk about as much today. This is a picture of my lab group. We have 3 graduate students, which are in the back row. I'm going to talk a lot about Mallory's research, and Mallory is here tonight. I'm not going to talk so much about my other 2 graduate students, but if you are interested to hear more about what we do, feel free to go onto our website and check it out. So after graduate school, I did even more training. I was a postdoctoral scientist at Woods Hole Oceanographic Institution. That was 3 more years of training in biological and chemical oceanography. And that brought me here to Skidaway Institute, and I'm excited to be here with you all. So I love this video when I'm introducing plankton. These are all kinds of plankton. This video was put together by French scientists from the Tar Oceans Organization and science communicators. If you are interested, you can check out the TED Talk, and they narrate it. But what I love about this is it's magnifying plankton. These are very small organisms that you need a microscope to see. They drift around in the ocean. My interests are in the phytoplankton. They're the ones that perform photosynthesis. When they come up, I'll show you. We're also looking at bigger organisms that chow on that phytoplankton as a source of carbon and energy. So phytoplankton, they come in different shapes and sizes. They can be quite small. This is one of my favorites. It's a diatom. Some have hardened shells. Some are more gelatinous. And they come in different colors as well, and that can be due to the pigments that they have to access light energy. So phytoplankton are also known as algae. We use those two words synonymously. All right, and today specifically, we're going to be talking about harmful algal blooms. So what sort of imagery comes to mind when you hear harmful algal blooms or toxic algal blooms? Let's hear thoughts from the audience.
Green stuff on top of the pond.
Green stuff on top of ponds. Yes, what else?
Red tides in the Gulf.
Red tides in the Gulf, totally. How do they smell?
Yucky.
Yeah, they don't smell great. And what about fish? What can happen to them? They can die, right. So these are all negative outcomes. Just last month alone, we've had algal blooms reported in several of our states. So we've seen them in Kansas, Utah, Florida, and California. These can be marine events, and they can also occur in freshwater systems like lakes. So I'm going to play a new segment that aired just a few weeks ago from Utah.
Yeah, you know, we've talked a lot about algal blooms, which are dispersed in the water. But this summer, some areas like the pond here in Liberty Park have seen algae mats. That's why they've got this caution tape and these signs of warning about animals, because they can be especially dangerous to animals and kids. In fact, the toxic algae is one thing being looked into after 30 wild ducks died here earlier this month. But tests are still pending, and veterinarians suspect other causes. Monitoring teams first started seeing the mats in Utah back in 2020. So we're still learning about them, and this is the first year that they've been found in the Salt Lake City area. Algae mats are different because they grow on the bottom and then wash to the shore in clumps, which is why it's important to keep kids and dogs away.
Problem with harmful algal mats is dogs really like how they smell, so they'll actually find them and eat them, which if toxins are present means the animal's going to get a really high load of toxin. And same thing with kids, you know, maybe they grab some accidentally, or they're playing on top of one and then drink the water. That's where the risk occurs.
Algae mats have been found in some...
All right, so these events are not great. Dogs can get sick, kids can get sick. They happen in freshwater lake environments. We're also going to talk about how they happen in marine systems. To understand more what's happening here, we're going to go over some basic aquatic biology. So in the surface of the ocean, we have our phytoplankton, and they take up CO2. So carbon dioxide is in our atmosphere, and it's in equilibrium with the surface ocean. So phytoplankton are taking up that CO2, and through the process of photosynthesis, converting it into organic matter. That's being shown here. A byproduct of that reaction is oxygen. So phytoplankton allowed for multicellular life like us to evolve. That organic carbon will typically sink to deeper layers in a water column, and then bacteria can go to town on that organic material. They're going to carry out the reverse reaction, which is respiration, where they consume oxygen and then reproduce that CO2. Mixing will bring that CO2 back to the surface ocean. So we call this the microbial loop because that carbon is being cycled. There is an amount of organic carbon that escapes that loop and makes it to deeper layers of the ocean. We call that exported carbon. So in this way, phytoplankton can move CO2 from the atmosphere as a gas to deeper layers of the ocean. And they can affect climate over geologic timescales by doing this process. And as I noted, phytoplankton, also known as algae. Right, so what is a bloom? Yes, we have a question.
I was just going to ask that question.
Phytoplankton are known as algae. Yep,
My
2 words.
question is, there's a line. How big is, there's not a line in the ocean, but there's a space in there where they affect each other?
Yeah, typically the surface ocean is where the phytoplankton biomass is because they're using that light energy. They're also using nutrients that are coming from down below. But we can talk more about this after the talk, if that's okay. Okay, so what is a bloom? These are natural events, and they're defined as a high concentration of biomass or the rapid rate of increase in biomass production. These happen naturally, so every year in the North Atlantic, there's an annual spring bloom, and that's where the phytoplankton are growing. The phytoplankton are the base of the food chain, and there's zooplankton that can feed on the phytoplankton. There's fish larvae and crustaceans that can then feed on them, and then there's bigger fish, all the way to the larger organisms. So these are productive events that happen, and they're so big that you can see them from space. And these are very important for that cycling and movement of carbon in the water column. These blooms depend on a number of different factors, importantly light and nutrients, as well as mixing. So what are harmful algal blooms? These are events that have negative ecosystem consequences. There have been over 900 poisoning events and close to 200 deaths in US coastal waters recorded over 50 years. And these can be economically damaging, with an estimated 75 million lost over a 25-year period. And this is from work put together by Don Anderson and others. So HABs can cause economic damage. They can directly impact ecosystem health by causing harm to animals, also known as wildlife, and humans. Specifically, let's talk about the modes of harm. Why are they harmful and what do they do? These events are characterized by toxin producers. So these are organisms that look like this that can produce toxins that can then ripple up the food web and affect larger organisms through the consumption. They can also be physical disruptors of animal tissue. We'll talk about some phytoplankton that have really long spines. Sarah has seen some of these, and they can hurt animal tissue that way. And then through oxygen depletion, which we just talked about, that bacteria consuming lots of organic material will consume oxygen, and that can cause negative consequences. There's a variety of factors that influence whether a certain bloom occurs, and it can vary by organism. So someone mentioned red tide earlier. That might be one kind of toxic HAB that can form. The frequency of these events is hypothesized to be increasing with global temperature. Some of these HABs like warm water. And so as we warm, there's certain niches or environments that are now opening up to organisms. So let's go through those modes of toxicity and see what they look like. The first one was toxin production. So here we have a dinoflagellate, which is shown with the tail, and a diatom, which is shown as that line. Diatoms are made out of silica, which is like glass, so they can be heavy. These organisms produce toxins, and they can be passed up the food web. So we might have a copepod that chows on these HAB species, small fish which chow on them, and then large fish, birds, and mammals which then eat those small fish, all that have concentrated that toxin. We also have shellfish, which are great at filtering water, and they can bioaccumulate and concentrate that toxin. So if a marine mammal comes along and crunches into that, it will be exposed to that toxin. And same for people. The next mechanism was Cattosaurus. This is a diatom, and hopefully you can see those long spines that it has. When there's a lot of them in the water, they're sharp and they can irritate fish tissue. And the last one was bottom water depletion. So in this schematic, you can imagine that there's phytoplankton at the surface. they're going to be using light and nutrients, and they can rapidly grow. So if you have a lot of material at the surface, some of that material, a lot of it, is going to rain down a little bit deeper in the water column, and then bacteria are going to be able to start consuming it and taking up oxygen in the process. This is going to result in what we call hypoxia or anoxia, which means low oxygen conditions. So this is bad news for organisms that respire. Fish can swim away. They can swim to up higher or try to get out of there because they need to breathe. But sessile organisms that are attached, they're going to suffer mortality if they're not getting enough oxygen. And depending on the wind, fish can get stuck in a channel of low oxygen. And this is how you get fish washing up on shores. Let's bring it back closer to home. Let's talk about coastal Georgia and nutrient sources. So on this island, we have the landings. I think we have some residents here today. The landings claims about 4,500 of the 6,300 acres on the island. They have about 6 golf courses. And we also have our institute here as well. So all that lawn and the golf courses require fertilizer, which are a source of nutrients and nitrogen to coastal waterways. It's estimated that there's about 170 pounds of fertilizer per acre used for lawns and golf courses in coastal Georgia. Another great source of nitrogen are hog farms. That's big in North Carolina, not so much in this area, but that can flush into coastal waterways as well. We know that high nitrogen loads are associated with the development of anoxic or toxic HABs because nutrients will promote the growth of phytoplankton. Something else to keep in mind is that we are on a septic system. I learned a lot about the septic system on the island from Bill Savage. He's one of our scientists here. And so we'll just kind of quickly talk about what that looks like. So our waste flows through pipes into these contained units where microbes consume that waste. They're anaerobic microbes, and they're performing what's called denitrification. They're turning nitrate from our waste into N2 gas. So that N2 gas is lost to the system. So it's a biological way to treat waste. Otherwise, like in cities, you have a sewage system. So we don't have that on islands. This is pretty popular on islands. So that waste goes into these units, and then some of it will overflow into lagoons where microbes can again continue this process. That extra material can get sprayed onto fields as fertilizer, and that's what happens. Septic systems generally work well and shouldn't lead to pollution, but if they're overloaded or they're not well maintained, they can, and they require a lot of maintenance. Their lifespan is anywhere from 30 to 50 years, and then they'll need to be updated. So here's a case study from Florida showing that septic systems led to nutrient enrichment of groundwater that made its way into coastal waterways and decreased water quality. Bill Savage has been measuring water quality on this island for a very long time, from the 1980s to the mid-2010s, and he has not seen any decrease in water quality, which is good news. That means our septic systems are performing well, but it is important to keep monitoring this to make sure that we're not seeing any signs of poor water quality. We also don't have quite as many people on this island as we do further in the Northeast, where population is much higher. Okay, so general consequences of nutrient loading or excess nutrients. They can lead to the accelerated growth of algae, which can promote hypoxia or low oxygen conditions at depth, and it can promote the growth of species that produce toxins. There's other ecosystem changes as well besides the HABs. You can have a shift in the type of phytoplankton that appear, and then that can change the type of predators that are present. So you can get changes at the ecosystem level. We're going to now take a look at some HAB species. These are some that we have seen here in coastal Georgia. The first one is Akashiwo. It's shown here. It's a dinoflagellate. It's this reddish brown color, and it's pretty easy to identify when we see it. It can turn water a brownish, reddish color, and it is one of the species that's known as red tide. There are others. Akashiwo in Japanese means red tide. It does not produce a toxin like some of the other ones that we'll talk about, but it does produce a sticky organic matrix called transparent exopolymer particles, or TEP. And this can coat the feathers of birds, which makes them not able to repel water, and they can't insulate. So they can go hypothermic. They also led to a mortality event in the Skidaway River Estuary, which we'll talk more about in just a moment. Alexandrium is another dinoflagellate. It's shown here. It does produce a toxin. Its toxin is called saxitoxin, and it's pretty nasty. It acts on the voltage-gated sodium channels of neurons and interferes with action potential or their ability to communicate with each other. What that looks like in people is paralysis, and it can lead to respiratory failure. It has been responsible for mass mortality events of fish from feeding on zooplankton that have concentrated this toxin. So I just wanted to now show you what Alexandrium looks like, just because they look a lot different than some of the other organisms that I work with. Alexandrium, it spins and moves at the same time, so it just looks like chaos when we're looking at it under the microscope, and it can form these chains, which I've seen, we've seen this off the coast of California. When we've seen it show up under the microscope, though, we've just tended to see a few cells, not in chain forming, or I haven't seen the chain forms. The next one here is a diatom. This is Catasaurus, which sounds like a dinosaur, but it's not, it's a diatom. It blooms off of the Pacific Northwest, and there it has irritated fish gills because it has these really sharp spines that you can see here. And there's nearly 400 species. It's a huge genus of diatom, and they have a little bit different morphology. So some have quite long spines and some are a bit shorter. The last one I'll mention here is Pseudonychia. I spent a large portion of my PhD working with this organism. Some species, but not all, produce a neurotoxin called domoic acid. And the ones that produce it don't produce it all the time. But it can lead to poisoning events for the humans and wildlife that consume it. Similar to the saxitoxin, it can make its way through the food web. This has traditionally bloomed off the West Coast, off of California. Recently, in 2016, though, it did show up in the Gulf of Maine, and it exceeded the federal action limit for domoic acid in shellfish, so it caused a closure of a shell fishery. So that was the first time we saw it there. So this was a pretty big deal, and so we're thinking that it's expanding in range now. Let's talk more about domoic acid. We are in the midst of a massive domoic acid poisoning event right now off of California. So this is from a news article from San Francisco, or sfgate.com. They did a great job covering this. There's dead sea lions and dolphins washing up on shore in very large numbers this summer. And so this is currently happening. This article was published just a few weeks ago. In this article, they state it's one of the largest in Southern California's recent history, with cases reported from San Diego to Santa Barbara. More than 500 sick and dying sea lions and at least 100 dolphins have washed up, but exact numbers are yet to be determined. This does happen historically. This has happened off the West Coast, but it has been 60 to 80 animals per year. In more recent years, it's gone up to 200, and then this year it's even more. The thought here is that there might be more upwelling happening off the coast of California, which will bring up nutrients to the surface. Diatoms are great at responding to that, especially Pseudonychia, which produces that toxin domoic acid. These blooms might be happening even further offshore than normal, which is why dolphins are being affected. So when this happens, NOAA, which is the National Ocean and Atmospheric Association, they'll put up signs like this to let people know not to go near the animals. When they've ingested this toxin, they are disoriented, they might be aggressive, they have bitten people. They also can have seizures, brain damage, and of course they can die. There are nonprofits like the Marine Mammal Center that respond to these animals and try to rehabilitate them. So this organization, once they get an animal, and they can't get to all of them, but the ones that they can get to, they'll give them fluids, a supply of fresh fish, an anti-seizure medication in some cases. They try to wait out the effects of ingesting this toxin. The animals that have ingested a lot of this toxin and over a prolonged period of time don't fare as well, but the ones that have only ingested a bit and more recently before they're captured tend to do better. So this is a nonprofit organization that you can read more about, and they do take donations if this is a cause you're interested in. Tying it back to coastal Georgia, let's now discuss HABs in our state. This is a map showing known HABs and algal poisonings in the southeast of the US. We can see HABs off of North Carolina, South Carolina, and Florida, but none officially documented off of Georgia. And HABs don't know state lines, so how do we explain this? A couple different points here to consider. One is that we are not monitoring to the same extent as adjacent states. We do a little bit of monitoring, and I'm going to talk about that in just a moment, but we haven't been quantitatively keeping track of cell densities. The second component here is that we have huge tides. We have 9-foot tides certain times of the month, and that's going to flush material into and out of our estuary. And then the last factor here is that blooms are ephemeral. These organisms will quickly respond to factors. They'll rapidly grow, and then they'll quickly dissipate. These matter to us on this island because there was a mortality event in 2017. So I wasn't here at the time, but Elizabeth Harvey and her team was. She was a scientist here at Skidaway. She's now in New Hampshire. And next door at the UGA Shellfish Research Lab, they did a routine water change for their oyster larvae, and the next day they died. And so Liz's team investigated why that happened, and they determined that there was a high number of akashiwo in the water at the time. Akashiwo doesn't produce a toxin that we know of, but it does produce that organic matrix that could clog the gills of oysters and cause mortality that way. NOAA has long recognized that monitoring is important to keep track of when these harmful algal bloom events are happening. And so they have developed these plankton monitoring networks. There are over 100 of these sites across 15 different coastal states. They also do them in freshwater systems as well. And Katie Higgins, who works at Marine Extension, has been overseeing this on the island for over 10 years. So these are citizens that are trained in identifying organisms, and they take nets into the estuary to concentrate phytoplankton and identify them under the microscope. Sarah Connor is one of them. We also have Norm here. This record is valuable for us in keeping track of qualitatively what organisms are showing up. There are about 12 different HAB species on the target list. I just have a few of them shown here, but the PMN volunteers, these are the ones that we really want to keep track of. So the first one we mentioned was akashiwo. It doesn't have a toxin, but it produces that organic matrix and can harm birds because it can coat their feathers, can also hurt oysters. We've seen that one in the Skidaway River as well as in the Chesapeake Bay. Alexandrium, that produces the saxitoxin. We've seen that in the Gulf Coast, the New England area, as well as the Chesapeake Bay. Dinophysis is one that we see, not a ton of it around here, but it has bloomed in the Gulf as well as in the Northeast. Karenia is also a big one. That produces the red tide that we hear about in Florida. So that's commonly bloomed in Florida. It's also been seen in New Jersey. Sudanicha produces domoic acid that blooms along the west coast. We're now also seeing it in the Gulf of Maine. And the last one here is Cattosaurus. This one doesn't produce a toxin, but it has those long spines. It can also contribute to low, hopefully we don't lose power. It can also contribute to low oxygen in bottom water. Okay, and so the data that the Plankton Monitoring Network volunteers have put together is useful for us in starting to understand seasonal trends. So what we're looking at here is their data over 10 years on Skidaway Island. On the x-axis, we have water temperature, and on the y-axis, we have the number of identifications. So in this column, we would have close to 50 identifications when water temperature was high or in the summer. I also have Sudanicha shown here in yellow, and then Dinophysis and Prorocentrum, they're identified to a lesser extent. So Akashiwo was an interesting one to us because they tended to identify it in warmer water. And Liz Harvey, she had a master's student, Liz Feiler, who looked into this as well as that 2017 event when it happened. And she concluded that the cause of the event was unknown, but it could be related to temperature, nutrient availability, salinity, and picoplankton presence. So an important factor here to consider is that Akashiwo can eat. It can photosynthesize, it can also eat. And so what it's eating is probably important in determining whether it's going to be abundant or not. By the same token, other organisms will eat Akashiwo, and that will consume them and draw down their population numbers. So these ecological factors will be important in determining whether a bloom forms. So given this background, we felt it was important to start monitoring for HAB species on the island with higher resolution than we've been able to do with the Plankton Monitoring Network alone. Socorro funded this project, and so now we are recording cell densities of HAB species across the year, and we're establishing their seasonal timing. The hope is that we can begin to document their relationship with water quality parameters. The big question is, when and why do blooms form? Mallory is the master's student that has been working on this project. She goes out and samples the estuary every day over the summer, sometimes twice a day, and once a week during non-summer months. And here's some of the parameters that she collects for. We, of course, look at cell counts, and we'll talk about how we do that in just a moment. She measures ammonium in the water as a source of nutrients. And with this YSI sonde, which has probes on it, we can get water quality, like dissolved oxygen, pH, chlorophyll A, turbidity, salinity, temperature, which is all very useful for us. And then we can go into public records to get an idea of factors like precipitation, wind speed, wind direction, and water discharge. So all of this could be important in the development of blooms. So going back to cell counts, how do we measure phytoplankton? There's a few different ways. One is microscopy, which is what the PMN monitors use. And this is the gold standard, because if you can look under the microscope, you can see it move, you can see how it swims, and that can help you ID it. We like to use fluid imaging with what's called a FlowCAM because it automates the process for us, but we don't get to see them move when we do this. And what I do, what my lab does is we do gene and RNA sequencing. And this gives you a fingerprint of these organisms with higher resolution than we can get just by looking at them. Some organisms you can't see as well, or you can't ID based on their morphology alone. In addition to what type of organism it is, we can also get at function. So what are they responding to in the environment? And that can help us understand when and why they bloom. Here's what the FlowCAM looks like. We have this instrument here on campus. There's a glass flow cell which particles rain through, and we have an objective to zoom in and a light source, and a camera takes images as the particles rain through. So here's what some of those images look like that come off the FlowCAM. And we can't image all of the particles, but we get about 40% of them. And once we have those images, we can sort them by shape and color, which makes it easier to count them. So the FlowCAM gives us community composition and quantitative cell counts. Here is a video that Mallory took of particles raining through the flow cell. And while this is happening, the FlowCAM is taking a bunch of pictures. And so we're able to zoom in and see what some of these particles are. This is a video that was taken by a summer intern a few years ago. Her name is also Liz. And I like it because you can see how Akashiwo moves. It spins while it swims. And when we see this under the microscope, we know pretty much instantly what it is. But the pictures look like this. So Mallory spends a lot of time scrutinizing these images to figure out what organism they are. So if we get them on their side or upside down, it can be a bit harder to ID them. But if we get a really nice front shot, it's easier to identify it. So Liz is up here on the left. She was our summer intern last summer, and she collected estuary data every day over 2 months. So we have Akashiwo cell densities here in purple. We have in yellow ammonium concentrations, and in green we have precipitation just from the end of June until mid-August. And in red we have temperature. So what Liz found is we went from essentially no Akashiwo to being counted to this explosion of around 60 cells per mil. And there seems to be a relationship with temperature. This is when the water got warm. So we thought this was interesting, but Liz had to leave. She graduated from UGA and went on to bigger and better things. But now with Socorro, we have a master's student, Mallory, who's able to sample more extensively. So Mallory started in February. And again, we have Akashiwo here in purple. Notice that the scale only goes to 14 cells per mil in this case. And we can see temperature began to increase as we get into the summer months. And by mid-July, we did see that increase in Akashiwo cells. It's not of the same magnitude as we saw last year. And in 2017, when they saw it get to 250 cells per mil, that was even higher. So it's clearly not just related to temperature. The extent of these blooms seems to be related to other factors. But we are going to continue monitoring through the season and see if we can capture some of those shifts now. Like when will Akashiwo begin to decrease? We've never been able to sample past August, but this year we will. Our vision for this project is that we can have a regional notification network to let people know when HAB cell concentrations are high. This could be for residents of the island and also for stakeholders that are in the aquaculture industry. And I can show you what our website is currently in testing phase, but this is on the Socorro website. And the idea is to load this with information and graphics about HABs in Georgia. And then this number is pulling from Mallory's spreadsheet that she updates every day of the week. So yesterday, or it's today, Mallory updated it. So today she counted 96 cells, which is about 24 cells per mil. So this is a way to get at HABs in real time. And looking ahead, we're interested in how coastal phytoplankton communities will respond to warming. So globally, coasts are warming, and we're curious what that rise in temperature will mean for HABs and how ecosystems might shift. So this is the first step in beginning to establish a baseline. With that, I'd like to thank everyone who has made this project possible, especially Mallory. She is the lifeline of this project. We also have the co-PI on the Socorro grant, Katie Higgins. She runs the Plankton Monitoring Network next door at Marine Extension, and she works with our PMN volunteers. We have Sarah and Norm here today. Neha Shah was our summer intern this past summer who's helped out with monitoring. Last year we had Liz Zink, and the year before that, Charlotte Green. So I'd like to thank Socorro for funding as well as support from UGA and Skidaway Institute. And thank you all for your attention. I'll take your questions if you have any. Yes.
I think what I'm still not clear about is, can these blooms shrink on their own? Do they disappear, or do they just like go forever? And is there anything you can do about them?
Yeah, they can shrink on their own. That's where the ephemeral part of this comes into play. So when we've seen them get to like 90 cells per mil, or 90 cells around there, the next tidal cycle, it might be half of that. So they can quickly go up and come down. But in Florida, red tide can persist for a while, which makes it especially harmful, especially where water is stagnant, like it's not flushing. But here in Georgia, we do have large tides, which promote a lot of that flushing. Yeah. Yes.
With the temperatures in the water in Florida, in some parts of Florida now, this summer reaching well over 100 degrees to levels that haven't been seen, and certainly not in recent decades. I guess I wonder what ability we have to predict how that will affect things. I mean, you're going to observe what's happening here, and that will help us establish what's really happening when the temperatures change here in Georgia.
Yeah.
Has there been enough data in Florida to get a feel for what is
Yeah.
happening with that increase in temperature? And you mentioned that clearly it's not just temperature, but other factors, and I wonder what factors those are, and to what extent the change in temperature will change the ecosystem and have an influence on other factors other than temperature.
It's a great question, and in short, we don't know exactly what the warming temperatures are going to do. We think they're probably going to change the ecology of the system. Certain organisms have a higher thermal optimum, meaning they like it warmer, so this can open up space for certain organisms to do quite well, and they can outcompete other organisms that prefer it cooler. Culturing is a great way to try to get at that. So if you can isolate these organisms that you're interested in, and we have a ton isolated, you can subject them to very warm conditions to see what happens. Do they grow better? Do they grow worse? Can they outcompete certain other organisms? In terms of what the factors are, for mixotrophic dinoflagellates that can eat and photosynthesize, the type of prey available to them will probably play a role. So is their preferred prey source available? And then who eats them? Are there predators around? If there's lots of predators, you're not going to have a bloom because they'll be grazed upon. So all of those factors matter. But the temperature part was interesting to us because Akashiwo clearly is not just responding to temperature. We've had a extremely hot summer, and we had a heat wave. And we're still not seeing them in the levels that they were a few years ago. So clearly there are these other factors, which is what Mallory and I are hoping to get at. Yes.
You mentioned about the golf courses, which I am a golfer, but I do worry about that a lot. I mean, how can we evolve into something that will be feeding the courses without it producing this kind of effect?
Yeah, well, at the moment, we're not seeing any evidence of eutrophication or decreasing water quality around the island. That's Bill Savage's unpublished work. We can talk more with him about that. But our septic system is probably working out okay considering the population it's handling. There are septic systems that are overloaded and not processing the sewage as they should. Septic systems should be able to prevent pollution from happening by the way that they work. So it is important to monitor, though, around these systems to make sure they are working efficiently. Yes. Yep.
So the patchiness is interesting to see, and how patchy each of the different species are. How much of that is, kind of a lack of connectivity among the populations versus there's some low-level amount of these guys everywhere, and these are the places that have the conditions that make them go haywire?
Yeah, it's a great question. We do see some biogeographical ranges of organisms. Like we'll see organisms at certain latitudes that just don't appear other places. But there's also a whole lot of everything is everywhere and the environment selects. We are seeing Pseudonychia now, toxic species, in a place where we weren't really seeing them before. So it's a little of both. Yes?
When harmful blooms are detected, particularly the ones that seem to persist longer periods of time, what sort of interventions have been tried and how successful are they? How much does it depend on species as opposed to the physical circumstances of the place?
Yeah, that's a great question. I'm not sure I've read about intervention strategies, but I'm sure there's been some brainstormed. What I understand is when this happens, you definitely let people know. So you're not going to eat any shellfish from a shell fishery that's been contaminated. And they treat animals as they show up. But in the lake systems, they'll put up signs of dogs and children, don't go near them. But intervention strategy is an interesting one. I haven't, nothing comes to mind in terms of what's been done. But I do know in places that are very populated, historically phosphates have come flushed into systems, like through detergents that can promote the growth of HABs. So regulating the amount of phosphates that you put in detergents and limiting the amount of excess nutrients that go into these systems can bring down those HAB events. But in terms of like an active event and how to stop it, there might have been strategies, but none come to mind.
Well, thank you very much, everyone. And remember our drop-in program in 2 weeks. Hope to see you then, and stay dry and drive safe out there.
Thank you.
Yeah.